following on from fertilisation, we're now going to have a look at a nice short one about what happens to this embryo following that process. So, about 4 to 5 days after fertilisation, the cell continues to undergo cleavage. And cleavage is a very fancy way of saying lots and lots of mitosis. And we can see that really clearly in the diagram. We originally have this fertilised egg, and this continues to divide from one cell into 16 cells by that 5th stage. What it will form eventually is known as a blastocyst, and this is after about 4 or 5 days. A blastocyst being a hollow ball of cells, and it has a cavity inside filled with fluid. So that cavity is this green space that I'm highlighting now. What we then have on the other side is what is known as the inner cell mass, and the inner cell mass I'll highlight in blue into here. The inner cell mass is made up of pluripotent stem cells, and they can differentiate into all of the different cells of the embryo. Around the outside of this blastocyst, and I'll try and highlight this very badly in orange, are what is known as the trophoblast. And this trophoblast will end up developing into the cells of the placenta. When we look at this diagram here, we can now see what starts off in the ovary, and I'll circle the ovary here, with our follicles. These follicles release that secondary oocyte, that unfertilized ovum, and on day 0, we're looking here at a process known as fertilization. Now, we can see fertilization occurring here as the sperm have actually penetrated through the zona pellucida, or the corona radiata, and the zona pellucida. Now, by that day 4 stage, we can see that originally fertilized cell has developed into a blastocyst. So what I'm writing up here is our blastocyst. It's really begun to develop. Now, the last little bit here is what we're going to be talking about in this lesson, and we can see that the blastocyst plants itself, or implants itself in that wall in a process known as implantation. So this is what we're going to be looking at for the remainder of this short lesson. So, implantation, what is it? Implantation, we know it's a process, and it's the process where that blastocyst begins to sink further and further and further into the lining of the endometrium. Now, the whole purpose of the blastocyst implanting within the endometrium is so that it can gain lots of nutrients, it can gain an opportunity for growth and development by taking all of the nutrients from the mother in order for it to grow. And this is what we've been looking at when we looked at the menstrual cycle, and we look at the build-up of that endometrium in preparation for this blastocyst itself. So there are some really important hormones involved during this time, and for that blastocyst to develop, it needs the endometrium to be really well maintained. So as a consequence of that, high levels of estrogen and progesterone from the corpus luteum are going to prevent the endometrium breaking down. Now, as these cells continue to grow and grow and grow, they will eventually develop into both the developing fetus, but also the placenta. And when the placenta has reached a particular stage of development, normally around 8 to 12 weeks, it secretes human chorionic gonadotrophin, which maintains that corpus luteum. Okay, and once the corpus luteum is no longer needed because the placenta is fully developed, it is going to keep releasing those hormones instead. Okay, so again, estrogen, progesterone released from the corpus luteum. They're going to continue releasing them thanks to the placenta releasing human chorionic gonadotrophin, which maintains the corpus luteum. At 8 to 12 weeks, it's then going to transition over to the placenta releasing estrogen and progesterone, and the corpus luteum can gradually degenerate into the corpus albicans.